Dear learners, welcome you all in the course of Immunology BZYET 141. Basically, it is a elective courses for BSc General. Dear learner, my name is Dr. Siaram. I am an associate professor, Discipline of Life Science, School of Sciences, IGNOU. Dear learner, today I want to discuss a very important topic that is known as the working of the immune system 4. Dear learners, my session is divided into two parts, part first and part second. In part first, we will discuss about the cytokines and properties of the cytokines, how cytokines communicate in the immune cells, how to classify the cytokines, and is the cytokines involved in any disease, and we will also discuss the different recombinant cytokines we can utilize for treating of the disease like cancer and SCB. In part second, we will discuss about the complement systems and the properties of the complement system, different components which are involved in the complement system, and there are the three pathways. One is the classic pathway, another is the alternative pathway, and third one is the manoj binding lectin pathway. There are the three pathways we will discuss in detail, and I will also discuss the function of the complement system and how can regulate the complement system. So these are the various things we will discuss in these lectures. What is the expected learning outcome of today's session? After completion of this session, you will be able to define the cytokines in properties and function of the cytokines. And after finishing of this session, you will be able to explain how cytokines differ in innate and adaptive immunity. And after finishing of this session, you will be able to list out the recombinant human cytokines and their uses, and define complement system and list out its function and pathways. I have already discussed it. There are the three pathways we will discuss in detail. And after finishing of this session, you will be able to describe various complement activation pathways and their regulations. So these are the expected learning outcome of today's session. So I have already discussed it. In part first, we will discuss the various things. In part first, it is cytokines. I think you have heard the name of cytokines during the COVID-19. Cytokines storm. It is a very important symptoms of the COVID-19 patients. So basically, storms of the cytokines will take place. Ultimately, death will occur. There are the certain drugs which are given by the doctors to the patients. Or ultimately cytokine storm will reduce, or they are able to save the life. The cytokine basically is low molecular weight, approximately 8 to 25 kilodalton soluble proteins. It is secreted by the different type of the cells such as the lymphocyte, monocyte and macrophages, and these cells, they are able to secrete different type of the cytokines, and cytokines act as a mediator of the cell to cell communication of the immune response. In case of immune response, various cytokines like interleukin 1st, interleukin 16, interleukin 12, the number of these cytokines are there. They can communicate with each other. Ultimately, immune response will take place. And cytokines regulate many critical biological process such as cell activation, cell proliferation and cell differentiation. After action of the cytokines, cell, it may be activate, it may be differentiate, or it may be proliferate with the action of the cytokines. It also determine the intensity and duration of the immune response. How much duration immune response will take place, 1 to 7 days, 1 to 5 days, 10 days. And cytokines also regulate the class switches of the various immunoglobulins, like immunoglobulins like IgG, IgM, IgD. The number of the cytokines, they will switch their classes. Which one is produced, it depends on the cytokines. Cytokines regulate the immunoglobulins, and switching of the immunoglobulins are there. And how cytokines, how do immune cells communicate with each other from the birth? The cytokine bind with specific receptors on the membranes of the target cells and triggering the signal transduction that ultimately alter to the gene expression up to the target cell. You can see in this figure, these are the cells. There are the inducing signals are there. These are the inducing signals. They can produce the cytokine. You can see the cytokines in red color. These are the receptor, and in this receptor, cytokines, they are able to bind it. Ultimately, signal transductions, they can give the signal to the cell. They can change, they can modulate the function of the cells are there. Ultimately, they can switch the immunoglobulins productions like IgA, IgG and IgD, IgM. So these are the very important functions of the cytokines are there. In general, cytokines and their receptor exhibit very high affinity to the each other. The susceptibility of the target cell to a particular cytokine is determined by the presence of a specific membrane receptor. So these are the receptor, specific membrane receptors are there which are present on the particular cell. They can recognize the particular cytokines. Ultimately, they can modulate their functions. What is the mode of the action of the cytokines? Cytokines have the three mode of action. One is the autocrine, another is the paracrine, and third one is the endocrine. So these are the three functions, three mode of action of the cytokines are there. The autocrine action, the cytokine bind to the membrane receptor on the same cell that secreted. You can see in this figure, these are the two cells are there. The cells, they can produce the cytokines, or it can bind own receptors, that is known as the autocrine. Here is the paracrines are there. Paracrines, cytokines bind to the receptor to a target with more near to the producer cells. So these are the cells are there. You can understand here is the paracrine. They can produce the cytokines. The cytokines, they can bind to the receptor with the other cells are there. So that is known as the paracrine. And third one is the endocrine. And cytokines travel through the through circulation and act as a target cells is the distance body part. So you can see in this figure, these are the cytokines, cytokine producing by the cell. It will circulate, or ultimately they can bind to the other cells. They have receptor, or their function of the cell, it will change. So these are the three mode of the action of the cytokines. Another thing I want to discuss, the properties of the cytokines. There are the several properties of the cytokines are there. Uh, Number one is the pleiotropy, redundancy, synergy, and antagonism. And cytokine can influence the expression of cytokine receptors. And cytokine regulate hematopoiesis. So these are the 7 very important properties of the cytokines are there. So I want to discuss one by one. Here is the pleiotropism. It means one cytokine can have the different effect on different cell. So you can see in this figure the cells, they shows the pleiotropy. It is activated cells. They can produce the interleukin 4s. They can bind with the different cell. It means one cytokine can have the different effect on the different cells. It can bind to the thymocytes, it can bind to the B cells, and it can bind to the mast cell. The mast cell, it will proliferate it. Here is the proliferation, activation, proliferation and differentiation of the B cells are there. So you can say it shows the pleiotropism. The IL-4 have ability to operate to the different type of the cell that cause the various biological effect. So you can say the one cytokines, they perform the different function on the different cells. That is known as the pleiotropism. Another is the redundancy. The cytokines can be redundant. Different cytokines have the same functions. You can see in this figure, here is the activated T helper cells. It shows the redundancy. They can produce the 3 cytokines like interleukin 2, interleukin 4, interleukin 5. They can bind with the B cell. Ultimately, proliferation of the B cell will take place. Here is the 3 interleukins are there. Their function is the same. There is no effect in the function. Like interleukin 2, interleukin 4, interleukin 5, it involved into the proliferation of the B cell, that you can say the cytokines, it shows the redundancy properties. Third one is the synergism, and cytokines can synergize with each others. The combined effect of 2 cytokines, interleukin 4 and interleukin 5, on the cellular activity is greater than the additive effect of the individual cytokines. So the synergies, here is the activated cell, you can see in this figure. Here is the interleukin 2 interleukin. They have the synergize, or ultimately the induced class switching of the IgE. So here is the B cells are there. They are able to produce the immunoglobulin E. They shows the additive effect. If one interleukin is produced like IL-1, it shows very slow production of the immunoglobulin E. And if the IL-4 it will combine, they will additive effect, or ultimately it will produce immunoglobulin E very fast, and the class switches will take place in case of the B cells. And fourth one, it is another properties of the cytokine. Cytokine can antagonize each other. It is just a opposite of the synergism. And cytokines such as interleukin 4 inhibit the effect of the another cytokines. You can see in this figure, it is activated T helper cells. Here is the interleukin 4s. If they produce the interferon gamma and block the class switches of the IgE induced by the IL-4. It means they show the antagonistic effect of the cytokines. So it is a very important properties of the cytokines. And fourth one is the cascade inductions. It is another very important property of the cytokines. The cytokine can stimulate a production of the other cytokines. It is a cascade type of the reactions are there. It is a multi-step feed forward mechanisms. And the cytokines interferon gamma secreted by activated T helper cells which bind on the surface of macrophages, and then after macrophages secrete the cytokine interleukin 12, they act on the another activated T helper cell and produce interferon gamma and tumor necrosis factor and interleukin 2 and other cytokines. So it is a cascade type of the reactions are there. One cells, activated T helper cell, they produce the interferon gamma. They are able to bind on the surface of the macrophages. It releases the interleukin 12. Interleukin 12 will bind again to the activated T helper cells. They produce the interferon gamma and tumor necrosis factor, interleukin 2 and other cytokines. So it is a cascade type of the reaction. It will maintain during the infections. After infection, the cytokines, they will produce it, or ultimately they will activate to the T helper cells and productions of the antibodies. The other properties of the cytokines, here is the properties 6. Cytokine can influence the expression of the cytokine receptors. Here is the receptors are there, alpha and beta chain are there. It is a receptor of the T cells. You can see in this figure. Here is the interleukin 4, interleukin 5 receptor transductions are there. It shows the up regulations, and if down regulations are there, transform growth factors beta, they will regulate the production of the cytokines. Means their expression, it would be very, very less during the down regulations. If up regulation, the expression of interleukin, it will very, very high. It will bind, or ultimately T cell will functions during the infections. In property number 7, the cytokine regulate hematopoiesis, the formation of the blood cellular components. The cytokines have many effects dependent on the target cells and the state of the differentiation, activation of the target cell, and presence or absence of the other cytokines. Here is the hematopoiesis into the bone marrow, means the formation of the blood cell in the bone marrow. There are the stromal cells. Here is the stromal cells are there. Interleukin first, they are able to bind it. They can produce the various factor like macrophages, colony stimulating factors, and granulocyte colony stimulating factors, and granulocyte macrophage colony stimulating factor. They can release it. They are able to bind with the stem cells. Ultimately, they are able to form the blood cellular component. So here is the role of the cytokines is a very important. They can regulate the hematopoiesis. There are the 7, I have already discussed the 7 properties of the cytokines. They can regulate the various function inside to the body after infections. Another thing I want to discuss the nomenclature of the cytokines. That there are the different type of the cytokines are there. The lymphocytes are also called the lymphokines. Monocytes, they can produce by the macrophages, that is known as the monokines. Lymphocytes, they are able to produce cytokines, that is known as the lymphokines. Leukocytes that act on the other leukocyte is referred as the intraleukins. Means one leukocytes, they can act on the different leukocyte, that is known as the intraleukin, like interleukin 6, interleukin 12, interleukin 2. Various intraleukins are there. They will act on the other leukocytes. And cytokine with the chemotactic activity is also called the chemokines. There are the nomenclature of the cytokines are there. We can give the different name of the cytokines which are produced by the lymphocytes, which are produced by the monocytes, which are produced by between the intraleukin and other leukocyte and other act on the other leukocytes, and cytokines with the chemotactic activity. So these are the nomenclature of the cytokines are there. Yes, another very important things I want to discuss, how can measure the cytokines inside to the body after infections. So we can measure it with the help of the sandwich ELISA. Here is very important techniques are there. Sandwich ELISA assay. Here is the aid cytokine conjugate. First of all, the cytokine bonds on the surface coated onto the bells and aid the enzyme conjugate secondary antibody. You can see here is the secondary antibodies are there which are linked with the enzymes. If you aid the substrate, ultimately color will develop. So we can measure with the colorimetry or ELISA readers. We can with the help of the reading like optical absorbance. Here is the concentration of the intraleukin in picogram per ml of your samples. So we can make a standard curve. Ultimately we can plot against the standard and unknown. So we can decide the concentration of the cytokines in particular samples. So what is the concentration of the cytokines? We can decide with the help of the sandwich ELISA. We can measure it the concentration inside to the body. Another is cytokines of the innate immunity. The number of the cytokines are there which are involved in the innate immunity, like interleukin first and interleukin first alpha and beta. Endothelial cell, macrophages and certain epithelial, they are able to secrete the interleukin first and interleukin first alpha and beta. And the cell target and biological effect, here is endothelial cell in the livers, activation, inflammation, coagulations and acute phase protein synthesis. So these are the function of the different interleukins of the innate immunity. And interleukin 12, it is produced by the macrophages and T cell, anti-inflammatory macrophages, antigen presenting cell, suppress interleukin 12 cytokine production as well as the expression of the co-stimulation and the class 2 MSC molecule. So these are the biological function of the particular cytokines like interleukin 10. And cytokines of the adaptive immunity, interleukin 1, and it is produced by the T helper 1 cells. It is a biological effect. T cells, NK cell proliferation activation, B cell proliferation, antibody synthesis. The role of interleukin is very, very important. It involves into the production of the antibodies. And interleukin 2 and T helper cell 2 cells, mast cell, NK cells, and the T cell, mast cell proliferation, macrophages, inhibition, interferon, gamma mediated activations, and B cell isotope switching, isotype switching of the Ig. So a lot of the cytokines which are involved in the adaptive immunity and innate immunity, but we are discussing very few. And cytokines and disease, the various disease which are caused by the cytokines, the bacterial septic shock which are occurred by the cytokines due to the several gram-negative bacteria, stimulation of macrophage by lipopolysaccharide, TNF-alpha first and interleukin first. And in this case, the drop of blood pressure drop in blood pressure and fever, diarrhea, systemic blood clotting in the various organ. Bacterial toxic shock, it is caused by the different toxins. In some infectious diseases like hepatitis, leprosy, and Chagas disease, and COVID-19, which are also involved in this case, they can cause the disease. In cancer, these are the various cancer are there: a hairy cell leukemia, wart, and Kaposi sarcoma viruses. It is caused by the cytokines. There are the certain recombinant human cytokines and their uses. In the recombinant human cytokines, the various recombinant in, cytokines we can utilize to treat the different type of the diseases like hepatitis. You can see in this table PEG interferon alpha 2A, Pegasus. It is a drug that used to treat the hepatitis C. In recombinant interferon alpha 2A, Roferon A, it is a brand name of the particular cytokines. Basically, it is a drug. We can treat the Kaposi sarcoma, lymph vessels lining, chronic myelogenous leukemia, and hairy cell leukemia, among the other disease. So these are the recombinant human cytokines we can treat the disease. And dear learner, I want to summarize part first. You have learned the various things related to the cytokines. The cytokines are low molecular weight protein that play critical role in cell signaling. And cell release them, and they have paracrine, autocrine, and endocrine activities. You have also learned the various properties including pleiotropy, redundancy, synergy, antagonism, and cascade inductions. You have also learned the cytokines may be categorized into chemokines, interferons, intraleukins, lymphokines, and tumor necrosis factors. And immune cells such as macrophage, B lymphocyte, T lymphocyte, and mast cell release the cytokines which involve the regulation of the immune response. And you have also learned about to the certain recombinant cytokines used to treat the various disease like cancer and like hepatitis C virus. Dear learner, we will take the short break. Don't go away. We will come within 2 to 3 minutes. Thank you. Dear learner, welcome in the part second. Complement system. It is a very important system are there. They can clear the infections. So basically it is made up of more than 20 protein that circulate into the blood and tissues fluid. It is discovered by the Julius Bordet in 1894, and it is a heat labile component in the blood. The complement system is non-specific antibacterial activity, and they are mostly made by the liver cell. So in the liver cells, more than 20 proteins they will form it. It will circulate inside to the body and tissues fluid, or they can recognize the particular pathogens, particular bacterial cells. You can say it is shows the antibacterial activity. It work in a series of the interactions, series of the cascades, series of the reaction which are involved in the complement system, each being the catalyst for to the next. It work for to the both innate and adaptive immunity. The complement activation is induced by the several innate immune component as well as the antibody bound antigens. And the complement cascade boosts phagocytes and antibodies ability to remove germ or injured cells and attack to the pathogen cell membranes and induce inflammation. So basically, after increasing the level of the complement system, inflammation will take place. The component of the complement system include the serum component. It is more than 20 proteins are there. The membrane regulatory protein and membrane receptors which are involved in this case. The main component of the complement system, there are the 9 central component in the cascade. They typically denoted by the capital letter C. The C means it is a main component. Complement components are there. It is denoted by the C, and these are the C1, C2, C3, C4, and C5, C6, and up to C9. So these are the various complement system components which involve in the different pathways. The complement component in a cascade, C1 has the 3 subunits, C1Q, C1R, C1S. The C1 it is divided into 3 groups. First one is the C1Q, C1QR, C1QS, and C2 and C5 have 2 components, A and B. The larger subunit are denoted by B, whereas the smaller subunit denoted by A. However, there is exception wherein C2A is the larger than C2B, and some component are designated by the alphabet V and D. So these are the several proteins are there, B, D, C, C2, C9. So these are basically the different components which are found in the complement systems. And multiple activation product like C3A and C3B, regulatory and inhibitory molecules are there like factor H and C4VP. These are the regulator and inhibitor molecules. And proteases, newly assembled enzymes like C4B2A and factor B, and effector molecule like C3AR and C5AR. So these are the various components which are involved in the complement system cascade. There are the more than 20 proteins are there. Ultimately, they will kill the pathogens. I want to discuss the complement pathway. These are the complement system pathway. It occurs by the 3 biochemical pathway. One is the classical pathway, another is the alternative pathway, and third one is the mannose binding lectin pathway. So basically in this case, antigen antibody complex will form, then the classical pathway will occur. Here is the lectin which are available inside to the bodies like mannose binding lectin, which are found in the bloods. They bind to the pathogen surface, or ultimately they will activate the complement system. And third one is the alternative pathway. Like pathogen surface, they have the different antigen like lipopolysaccharides, teichoic acid, number of the molecules which are present in the which are present on the surface of the molecule. They will activate the alternative pathway. So they can activate the complement pathway and recruitment of the inflammatory cells, and opsonization of the pathogens are there. So ultimately your cells, they will phagocytize with the help of the macrophages after binding of the some complement proteins on the surface of the pathogens, and killing of the pathogens. Like they can form the membrane attack complexes like MAC formation. They will kill the pathogens. So these are the three pathways are there. I want to discuss one by one. First one is the classical pathway. You can see in this figure, it is a pathogens. Here is the antigens are there. It is antibodies like IgM and IgG. They can bind it, and their FC site, and different proteins like C1, C2 and C4, they will bind it, or it will activate the pathway. Here is the classical pathway. Antigen antibody complex trigger the classical pathway, and three component C1, C2 and C4 can make complexes with the antigens. Here is the antigens are there. They can make a complexes, and IgM and IgG bind to the antigens as they enter into the bodies. It causes the conformational change in the antibodies FC region, exposing to the binding site for to the C1 component of the C1. So C1 they can bind with the FC region of the antibodies, and the C1 is the large multimeric protein, consists of to the 1 C1q and 2 C1r and 2 C1s molecule subunit. C1q interact with to the FC regions of the antigen bound antibodies, and C1r and C1s protease can be cleaved to the C4 and C2. Here is the reaction is very important. They can bind with the particular molecule. Ultimately they can make a cascade of the reaction of the complement systems. And C4 is divided. You can understand here is the C4 is divided into a smaller C4a and larger C4v unit. So it is divided into two units. One is the C4a and C4b unit, when the immune complexes linked to the C1, and C4a diffuse away. C4a it will diffuse away, but activated C4 attached to the target surface at C1q and attract C2, and which is cleaved into two units, C2a and C2b. The bigger unit C2a, here is the bigger unit C2a, and form to the C4b C2a complex with C4b, while the smaller subunit C4b diffuse away. C3 engage when the C4, here is the C3 are there, it will engage when the C4b and C2a is activated. So these are the cascade type of the reactions which are involved into the complement systems are there. Ultimately the MAG will forms, or they can kill the pathogens. And it is cascade. I have already discussed it is a cascade type of the reaction. In this cascade, the C4b-C2a complex is known as the C3 convertase. So here is the C3 convertase are there. C3 convertase, because it is responsible for converting C3 into the active form by degrading C3a from larger subunit C3b. Here is the C3b are there. Here is the C3. It will diffuse away. And C4b C2a can cleave large number of C3 molecule with the single molecule, and C3b form to the extremely stronger bond with C4b C2a, and some C3b molecule do not interact with to the C4b C2a. It enhance the phagocytosis and opsonizations. Here is a very important reaction which are involved, and it will enhance the phagocytosis and opsonization after binding with the particular complement system proteins, and the macrophages, they can engulf the pathogens after binding of the particular complement systems, and C3 activate mast cell and causing the histamine to be released. So you can see it, there is the cascade type of the reactions which are involved in the in this pathway. And when C3b bind to the C3 convertase, it transforms to the C4 BC2 AC3b, which also known as C5 convertase because it activates C5. So here is the C3 convertase and C5 convertase, which are very important. They can maintain the pathway, the cascade of the pathway of the complement system are there. And C5a diffuse and C5b is preserved by the adhering to the C6, and C5b C6 then connect with to the C7, and the C5b C6 C7 complex is injected into the cell membranes and phospholipid bilayer, where it interact with to the C8, and C5b C6 C7 C8 complex activate C9 and resulting into the formation of the MAC are there. So you can see it here is the membranes are there, the bacterial cells are there, it is a different type of the complement proteins are there. They can bind to each other. They are able to make a pore inside to the bacterial cell. Ultimately the cytoplasm, and they will release it. So these are the membrane attack complexes are there. They can insert inside to the body of the bacterial cell. They will release the cytoplasm, and other materials are there, ultimately kill the pathogens. And osmotic stability of the cell is compromised. The lysis ensues as a result of the influx and water and electrolyte loss. This particularly effective of the gram-negative bacteria, since the development of the MAC is outer membrane, and these bacteria is quite straightforward. It is difficult to case of the, in case of the gram positive bacteria, the presence of the rigid thick wall, it may be difficult to kill the pathogens. So means you can say these are the very little limitations of the complement systems are there. Another is the alternative pathway. It is activated by the cell surface. Antigen include lipopolysaccharides and other antigens which are present on the surface of the bacterial cell, which initiate this pathway, that is known as the alternative pathway. It does not require antigen antibody complexes for initiation of the alternative complement pathway. If the bacterium attached to the host cell, you can see here is the component of the microbial cell surface. They have the particular antigens are there, like lipopolysaccharides, C3, they can bind it, it will remove away C3b. Here is the different factors are there, like factor B, factor D, and here is the A will release, and B factors and preproteins, they will bind together. They can form the C3 convertase. Again here is the C3b. They can form the C5 convertase. and the C5A is released and C5B involved in this reaction. Here is the C6, C9, they will combine it. Ultimately, they can form the membrane attack complex, or ultimately they can kill the pathogen. So these are the alternative pathway of the complement system. Here is C3 molecule immediately connect to the antigen and activate it. And C3 component undergo the spontaneous hydrolysis and yield to C3A and C3B. The B, D and P factor interact with to each other and bind to the C3B and produce a C3 convertase. C3B bind to the antigen and subsequently bind to another serum protein known as the factor B. And factor B revealed to the location on the serum protein D and serve as the substrate and enzymatic activity form of the proteins. And factor D break down into B and VA and VB, resulting into the formation of C3 convertase. Here is the C3 convertase you can see in this figure. And as per classical pathway, C3 convertase create C5 convertase which form the MAC. Here is C3 convertase and C5 convertase. They are able to make the membrane attack complex. And C3B and VPP can activate more C3. Consequently, the pathways are frequently called to the amplification loop. The loop is enhanced the presence of the bacterial and fungal cell wall, but inhibited by to the chemical on the surface of the normal mammalian cells. Another is the lectin pathway for or manoj binding lectin pathway. You can see in this figure, here is the membrane manoj binding lectin or ficolin. So these are the very important components which are found into the bodies. And when the bacteria attack to the host cells and the MBL, they can recognize the manoj which are present on the surface of the bacteria. And here is the membrane associated serum protease 1 and serum peptidase 1 and 2. They involve it, or ultimately it will react, or they can form to the C3 convertase. The Salmonella Listeria can activate the complement system without having antibodies and endotoxins. And some fungi in viruses, HIV, have ability to activate the manoj binding lectin pathway. The manoj binding lectin concentration rises dramatically during the inflammation, indicating body to responding to the stimulus. Manoj residues are to recognized by the bound by the circulating lectins. So these are the cascade type of the reaction which are involved in this pathway. Here is the manoj binding lectins which are available inside to the body. They are produced by the they can recognize the pathogens, they can bind it, or ultimately they will activate it. When the manoj bind to the manoj binding lectin bound to the manoj residues on the pathogen surface, the manoj binding lectin associated serine proteases MASP1 and MASP and manoj associated serine proteases are activated. Both it will activated, and manoj associated serine proteases first and second cleave C4. They are able to cleave the C4 and C2 to form C3 convertase, and C4 B, C4-2A, which are analogous to the tetrameric complex formed by to the C1R and C1S. The procedure now proceed to the make C5 convertase and finally make the MAC membrane attack complexes, and did it the previously. So there are the three pathways which are involved in the complement system, or ultimately they can kill the pathogens by the attacking on the membranes and opsonization process, and various mechanisms which are involved for to the killing of the pathogens. There are the two after that there are the two pathways are there. First one is the lytic pathway, splitting C5 and attaching C5 to target kick start to the lytic pathway, complement C6, C7, C8, C9, then join C5 to form to the MAC, and MAC can kill the bacteria by lysis and then inserted into the outer membrane. So these are the mechanism which are involved into the complement system, or ultimately they can kill the pathogens. So these are the series of the proteins C1 to C9. They can join together, they are able to form to the membrane attack complexes, or they are able to kill the pathogens. It is role in the C3 into the complement pathway. First step, differ into the each of the three pathway. All of the route where the create enzyme complexes such as C3 convertase, which cleaves C3 to C3A and C3B. C3 play an important role into the process by keeping to the complement cascade alert. It is very difficult, it is very important point to convergence of the three pathway. It help to the amplification up to the complement response. It also help to the better coordination of various immune responses by exerting direct effector functions. C5 convertase are formed when C3 attach to the C3 convertase, and C5 convertase cleaves C5 into C5A and C5B. The activation of other complement systems component by C5 convertase, which is produced through the alternative classical and lectin pathway, result into the development of to the membrane attack complexes which kill the pathogens. What is the function of the complement system? So there are the five functions of the complement systems are there. One is the chemotaxis, second is the opsonization, and third one is the cell lysis, and fourth one is the activation of the mast cell, and fifth one is the immune clearance. So their complement system which are involved into the killing of the pathogen by the several mechanism like chemotaxis, opsonization, cell lysis, activation of mast cell, and immune clearance. And disease related to the complement system, these are the various diseases are there, like erytheromatous. It is deficiency, basically it is a autoimmune diseases, the deficiency of C2 and C4 complement system. Pyrogenic bacterial infection can be caused by the lack of the C3 and D factor. C9 and C5 and C9 efficiency deficiency, you can say it is a MAC deficiency can result into the Neisseria. Infectious such as to the gonorrhea and meningitis is due to the complement system. Due to the lack of regulatory protein, complement and site become two. So these are the various diseases which are involved into the complement systems. And here is the regulation of the complement system. It can be regulated by the number of the cell membrane in plasma protein. The high quality of the sialic acids in the membrane of the most mammalian cell cause the complement inactivation. So it is a regulation of the cytokines by the two methods. One is the number of the cell membrane into the plasma proteins which are involved into regulation of. Another is the quantity of the sialic acids in the membrane, and most mammalian cell cause the complement inactivations. Dear learner, I want to summarize my session, part second. I have already discussed it, the complement systems, and it is more than 20 inactive protein that circulate into the blood and tissues fluid, and it perform is in the form of the cascades. I have already discussed it. It is a chain of the reactions of the complement systems are there, and each acting as a catalyst part to the next. In the complement system, activation occur by the three biochemical pathway. So there are the three pathway are there. I have already discussed it, like alternative pathway, classical pathway, and MBL pathway. Dear learner, if you want to learn, if you want to clarify a number of the things, you can go through the IGNOU SLM B, BZYCT 141 Immunology courses, unit number 12. You can read it, you can clarify. If you have any query, you can mail me on the this mail like cram1@ignou.ac.in. Thank you.